Now that we have learned the basics of protein structures and how these structures are resolved, let's take a look at the information we can gain from each protein structure. Specifically, this section will outline the detail with which we can gain biological understanding from protein structures. First, let's take a look at this CUS-BA efflux pump. CUS-BA efflux pumps are resistance nodulation cell division transporters, or R&D transporters, responsible for expelling toxic compounds from the cell and associated with virulence and drug resistance. This pump expels silver and copper ions from the cell, compounds that are often found in topical antibacterial creams. To combat the effectiveness of these pumps, scientists must understand how these structures function. Until recently, these structures were difficult to resolve in their native conformation because they span multiple membranes. With cryo-EM, these structures have been resolved, and the physiochemical properties associated with oligomerization or ion binding can begin to be probed. Quickly, pause the video and read the detailed accounting of this structure from the excerpts on the left. Notably, the transport mechanism of metal ions through this transporter is still not fully understood. The bottom excerpt shows how by mapping physiochemical properties to the resolved structure, specifically the charge of amino acids, can help elucidate hypotheses for this mechanism and may allow for educated hypotheses how to combat these bacterial defense mechanisms. Since we are facing a pandemic, let's quickly look at some viral proteins and how structural biology has elucidated their mechanisms of infection and helped guide treatment development. Here, we are looking at cryo-EM images of the H1N1 flu, responsible for the 2009 swine flu outbreak. Below, figures C and D show the hemagglutinin called HA. This is the most abundant virus surface protein of flu viruses. These proteins attach the viral capsid, shown below in gray, to the host cell at the top, red. So what we're looking at is this blue that attaches here to the viral capsid, and this red part of the protein which attaches to the host cell. The host attachment area is highly variable between different flu viruses, making the creation of antibodies that target this red region difficult and often ineffective against multiple flu strains. The stem region, however, is highly conserved, making this a great target for the development of antibodies. However, it is not known if this region is accessible to antibodies because it is buried towards the inside of the viral capsid and possibly shielded from these antibodies. Using cryo-EM, the binding of C179 antibody to HA was explored. C179 bound HAs image in a different pattern than unbound HAs, as shown on the right, in the differences between the blue and green images. Using the images of the entire H1N1 capsid, it was found that broad-spectrum C179 antibody binds to 75% of the HAs on the viral surface. This demonstrates that vaccines that elicit such antibodies may soon have universal effectiveness. Here, we can take a detailed look at the cryo-EM structure of the HIV Envelope Trimer. This protein is exposed on the virus surface and is involved heavily in membrane fusion and infection. The membrane proximal external region, or MPER, is, as the name suggests, the region just external to the host membrane. This region is highly conserved and is of great interest in developing drug targets. If host antibodies can bind to this region prior to HIV protein fusing with the host membrane, infection may be mitigated. In the top two figures, antibodies targeting this region are shown in blue and pink. The HIV envelope is highly decorated in glycans that stabilize the trimer pre-fusion. These are shown in the green on the left. Cryo-EM identified new glycans in this study. This study also modeled glycans at positions 88 and 625 to be less mature than native glycans, meaning that they were shorter than the native glycans. In the center figure, glycans at positions 88 and 625, these green circles, which I have circled in red, are in close proximity to the antibody MPER binding site, shown in gray for the antibody and yellow for the MPER. This suggests that fully mature glycans at these positions may provide steric hindrance and prevent the antibody binding to this membrane proximal region. Further evading drug target, the MPER demonstrates flexibility as it is targeted by antibodies. When the antibody 10E8 on the bottom left, or CD4 center right, are bound to the envelope protein. The protein is lifted out of the membrane compared to its native state, shown in the middle. Interestingly, the binding of CD4 and the opening of the MPER trimer, shown here, relieves some steric hindrance of the glycans and allows 10E8 to bind more effectively, shown on the far right. The intricacies of this dynamic binding and conformational changes found in MPER binding events can help scientists develop antibodies to avoid these molecular roadblocks in neutralizing the HIV envelope fusion. Questions such as, should the antibody be designed to be longer and more flexible to accommodate binding during the separation event, or should the antibody receptor shape be tailored to avoid the possible steric hindrance at positions 88 and 625, can begin to be answered, and that's all thanks to structural biology and cryo-electron microscopy. As we quickly approach the present day, a quick structural biology dive into the ZIKA virus of 2016 is worthwhile. Very briefly, this study resolved the structure of the ZIKA virus and found that the amino acid sequence is similar to other flavivirus structures, except for 10 amino acids at the asparagine 154 glycosylation site shown here. This suggests that antigenic variation at this site may be responsible for differences in virus transmission and disease. The West Africa Ebola outbreak of 2013 to 2016 was the largest Ebola virus outbreak ever recorded, killing over 11,000 people in 28,000 cases. Although the number of deaths are significantly less than many other milder infectious diseases, the kill ratio of this virus remains one of the highest. Briefly, the Ebola life cycle begins when its glycoproteins are recognized by a host cell, which initiates Ebola's endocytosis. Once free from the endosome, the nucleoprotein, shown in red at the bottom left, relaxes and exposes the viral RNA for transcription. The viral components are translated and the capsid is reassembled, leaving the cell. In the center, we see the nucleocapsid, which is in large part made up of oligomerized nucleoproteins. The unit is shown in blue. These structures bind to viral RNA and shield it from degradation, shown in red. Comparing the monomeric state to the resolved EM structure of the nuclear capsid, a model containing the proposed conformational changes required during the oligomerization and viral RNA binding was created, shown here on the right. Important residues and helices in the nucleoprotein were identified, suggesting that these binding pockets could be potential drug targets. Since we are conducting this lecture online due to the coronavirus pandemic, it is worth taking a look at the structural biology advancements in understanding this disease. The family of coronaviruses uses proteins called spike proteins to mediate entry into host cells. Spike proteins recognize host cell receptors and facilitate the fusion of the viral and host membranes. Depending on the species of coronavirus, the S1 domains of these spike proteins can vary, allowing them to recognize different human receptors. On the right hand, different coronavirus human receptor targets are shown. Notice that SARS and MERS, which are responsible for outbreaks in 2003 and 2012 respectively, are also coronavirus. More notably, notice that SARS binds to the human receptor ACE2. Fast forwarding to the issue at hand, by the way, you should probably wash your hands after this lecture. On December 31st, 2019, China officially reported its first case of novel coronavirus, called COVID-19. 12 days later, the first confirmed death due to COVID-19 was confirmed in China. On March 1st, the US reported its first death. On March 11th, the WHO declared the coronavirus a pandemic. As of today, March 12th, 2020, there are over 101,000 confirmed cases of COVID-19. On February 19th, 50 days after the first reported case of coronavirus, the structure of the important spike protein of COVID-19 was solved and published in Science. More interestingly, the structure in its bound state to ACE2 demonstrated that COVID-19 binds human receptor ACE2 with 10 times greater affinity than the SARS coronavirus. Additionally, 3 antibodies known to bind SARS coronavirus were tested against the COVID-19 protein without success. On March 4th, the group on the right demonstrated the structural detail of ACE2 binding to the receptor binding domain of the SARS spike protein. Understanding the binding event at the angstrom level may facilitate the design of decoy ligands or neutralizing antibodies for these spike proteins.